So now we get to a very exciting part of the day, my favorite. We get to talk about drugs, which I love, drugs. But this is the time we get to learn all about the exciting things that are coming down the pipeline for FSHD therapy development, to share all about the drug development and the clinical trials. I'm gonna have Dr. Korngut come back up. And after he's spoken a bit about the drug development and clinical trial overview, we're gonna have guest speakers from Avidity, from Satellos, from Histone Therapeutics, and from Solve also be speaking. And then Dr. Korngut is gonna host a panel with all of these drug companies. So take it away. Are you recording?
Oh.
Hang on.
I am recording.
You are?
Yep.
Okay, we're recording.
Okay, hi everybody, it's a wonderful day. So what I'm hoping to share right now is a little bit about how clinical trials work, and then just give some updates of what kind of drugs, technologies are being developed to treat FSHD. And in neurology, we think of two major categories of treatments. One are symptoms, how do we help with symptoms of a given disease? But this is exclusively focused on disease modification. So how do we preserve muscle? How do we reduce the actual cause of this disease? And so I'll be speaking about that. It's impossible in 15 to 20 minutes to really do this topic a whole lot of justice because there's a lot of detail here. So I'm gonna just kind of gloss over, but I hope what you come away from this is a sense of cautious optimism that the field is heading in the right direction, that there's a great deal of momentum. And I think what you'll see is that there is reason to be optimistic in this day and age. So if you look at this slide, it's a little bit busy, but you can see, yeah, it's my cursor. And so of course, FSHD is caused by, from a genetic perspective, by a reduced number of P4Z4 repeats. Which then sort of releases a protein which should not be present in muscle. So in the healthy state, there is no DUX4 protein in the muscle, but here you have, because of these reduced repeats, you have expression of DUX4 protein, which then causes an immune reaction, causes toxicity to muscle, causes breakdown of muscle, which we are all too familiar with. And I think what I like about this slide is that it shows that across these different steps within the disease, at each of these steps, there is a company and a specific treatment to address that step that is being developed. I gave a similar talk about 10 years ago now about a disease called spinal muscular atrophy, where we had 3 drugs in clinical trial development, similar to this stage. And I thought, well, that was exciting. But, you know, here we have a lot more. At this point in SMA, all 3 are on the market and have really transformed individuals' lives with that disease. And of course, that's what we are, you know, hoping for here. But I think just the sheer diversity of the different targets that are being addressed, and we'll go into a little bit of more detail about these specific treatments that are being developed. Importantly, none of them are proven. So this is kind of a frustrating part, of course, where you can sort of see this work go on, but it's not here yet. And again, it's really important that we go through that whole process. And so therapeutic development, so the process of developing a treatment is quite a long one. On average, can take a decade, if not longer. I think the average cost to develop a drug is on the order of $1 billion. After all is said and done, I think on average. Now, I think it's less in rare disease as a whole. But you know, I think this all starts with understanding. It's hard to make a treatment for a disease if you don't know what the mechanism of the disease is. And as you saw, there's quite good understanding. It's not perfect, and there's, you know, lots more to uncover there. But understanding the disease, understanding how the disease progresses, understanding all of those different components of establishing animal models so that you can test different therapeutic strategies. You know, the MOVE study, I think, is a great example of a study where they so deeply phenotype and characterize individuals over multiple time points. So that's an important example of understanding what is the natural history? How does this disease progress without any treatment? Because if we can't understand that, it's hard to understand if the treatment's making a difference to that natural history, right? And these things are all easier said than done because everybody is unique. Different individuals are diagnosed at different ages, have different progression rates. And this is a major challenge in FSHD, just like many of our neuromuscular conditions. Ideally identifying biomarkers, so these are blood tests or other tests, MRI imaging, etc., that can sort of give us a glimpse of where is somebody at and can this treatment help them, and is there a way to kind of shortcut giving this drug for a year and a half? So all this research sort of goes on at once. One thing we're very involved has been trial design, is understanding how do we optimize the way we do clinical trials to make them more accurate, make them more efficient. And then of course, clinical trials are really necessary because once a treatment, let's say a molecule or a genetic treatment is identified, safety comes first, right? So as clinicians, when we recite the Hippocratic Oath, first we do an alarm, and that really is the basis of this. When you many of you may have studied ethics, and there's a sort of sordid history in human history of unethically conducted experiments, and so there's very good safeguards in place and very appropriate accountability for doing any experimental research. And so any clinical trial in North America and in Europe does require ethics committee approval, does require quite rigorous oversight. I will say there are places in the world where they will offer clinical trials and they'll charge you for it. Those would not consider to be clinical trials, that's just marketing. And so in the setting of a clinical trial, you're the volunteer. That is what you are contributing, which is a lot, right? And so I think that's an important distinction. And then really trying to understand, so you can imagine if we have a couple of hundred people with this disease, everybody looks different, everybody's progressing slightly differently. You're going to get a lot of variability. And so whenever we do a clinical trial, we do need a placebo group to understand, is the treatment doing something versus if somebody did not receive the treatment. And importantly, we randomize between those two groups. And again, some people find this frustrating, which is completely understandable. The problem is, we as the investigators want you to be better, you want to be better. It's a very challenging way to understand if a treatment works, even if it's very effective. And we've certainly, when I look back on our trials that we've done, I always go back and sort of say who I thought was on drug and who I thought was on placebo, and I'll go 50% right. Meaning, I don't know. Maybe I can't tell. So which is a good part of the design, I think. And so then, again, our regulators are involved. So I think the number one thing, as I mentioned earlier, is that, safety of the participants is critical, and making sure that we're doing the right drugs, right? That we're not doing drugs that have a 1 in a billion chance of working. We're doing drugs that make biological sense, and that there's data from this earlier part here, from preclinical research, et cetera, to suggest this could be helpful, right? Because otherwise it's not to just do a trial. And so there's the 3 phases that I'm sure you're familiar with. You can see that, this usually is around 64, around that number of subjects. These can sometimes be healthy individuals, sometimes have the disease of interest, depending on the design. And so here we're assessing, is the drug safe? Is there side effects that we need to know about? If all 60 people start getting into kidney failure, this drug's not going to get very far. And trying to understand how the drug is absorbed, et cetera. Phase 2 is now, is there more, accumulating more safety data? Is there some preliminary sign that there's benefit, that the drug could be effective? So importantly, if a phase 2 smaller clinical trial shows a benefit, doesn't necessarily mean there is a benefit, because often the sample size is so small, you can just get that by chance. So if the treatment group progressed slower just by chance, it's gonna look like the drug doesn't work. And so whenever we have our two groups, we always say ideally we would have 100 clones of, you know, if we had 200 clones of the same person, we put them into two groups, because then the only difference is the treatment and the placebo, right? Of course, that's not how life works. And so then the phase 3, again, number one, is it safe? How effective is it? And is there a benefit to the patient population that we're studying? And so really it's the phase 3 that tells us the answer of whether there is benefit. And so now the challenge is, of course, this takes years to work through these, but part of that is what we don't want is potentially harmful drugs that are not helpful to you. So that's important. But we also want to protect the participants in these trials by, you know, if you're participating in a phase 2 or phase 3 trial in particular, that there's as much safety, you know, understanding as possible at that time point. So it's kind of reducing the risk for everybody all along, right? And of course, that takes time. And so the Fulcrum trial, we saw some data from, so I'm not gonna belabor this again. Dr. Wang had shown that. But of course, this was a small molecule, so a chemical entity that suppressed P38, which we know interacts with DUX4 expression. And so, you know, unfortunately, the trial was completed and didn't show a positive result. So there's no difference between the treatment and the placebo. We saw the curves before comparing the two. But I think importantly, the way I always think about it is we're not only assessing the drug, which I think there's lots of learnings here about what happened in that clinical trial, but we're also assessing how many patients were in the trial, what kind of patients were in that trial, how long, you heard reference, maybe 48 weeks isn't enough. Is reachable workspace the right outcome measure in terms of all of that information? So there's a massive, you know, it's not like trial's negative, forget about it, let's just do this all over again. It's how much knowledge can we extract out of this so that the next phase 3 is designed more intelligently based on this result. And so this was a pretty large trial, 260 patients, phase 3. We I think had 5 or 6 patients in Calgary as part of that trial. And then, yeah, so then just to highlight, just think of a couple of examples, and I added this photo here of this cow with very large muscles. And of course, we're not cows, obviously, but genetically, we're not all that different from the animal kingdom. And this is an interesting story because I think it sort of explains a bit about how these findings kind of make it into the clinic in terms of clinical trials. Because these cows have huge muscles, and the reason they have huge muscles is they have a gene called myostatin. We all have a gene called myostatin that turns off muscle growth. And that's how we help balance how big our muscles are. And they have a mutation in that gene. So that, and these are specific Belgian beef cows that have this mutation. And so their muscles overgrow. And so this led to a whole line of research and ended up with a couple of different treatments looking at this. And of course, they're not interested in curing the cows per se, but trying to apply this, you know, how can we help individuals with diseases? And so these anti-myostatin agents have then been developed to treat myostatin in the blood that impacts muscle to help you know, build muscle bulk and strength. And this is being looked at across different diseases, including FSHD. And so this is a phase 2 trial that's ongoing of 48 participants. And so again, that's a very different approach than the small molecule that Fulcrum was developing. And I think, again, if there's any message to take away, it's not the exact details or the super muscular cow, right? I think the message is that there's a lot of interest in this whole pathway all the way along. which is what, as a community, what we want, right? So if I look at other neuromuscular communities, there's way less research, way less clinical development, way less therapeutic development going on. And different reasons for that, but certainly at this point, there's a tremendous interest and investment of effort and resources. And so this trial, this anti-myostatin, it completed recruitment. We're expecting data in the third part of 2026, so next year. And so we should have some more information. So as you see, like that is a long way off, but overall, as you'll see, these readouts will happen, and we'll get more and more information over time. Another study, this is a smaller study of 40 participants. Again, Roche drug Satellos, which blocks a certain part of the immune system, and we know there is some element of immune involvement in FSHD. And so this study is running as well, but again, a completely different mechanism of action again from the other. And this one's fully enrolled, so by mid-2026, we'll have some more information about that. I know we have a colleague from Avidity joining for this session, so I'm going to just skip this component because she will do a far greater job than I could. Arrowhead, again, so we have small molecules, we have these different platforms for accessing muscle in terms of therapeutics. And so in FSHD, the goal is to turn off this expression of DUX4 protein, which should not be there in the healthy state. And that part of the challenge with these treatments is targeting muscle. It's how do you get the treatment into the muscle and avoid getting it into the heart, the liver, the other areas that's where these off-target side effects can come from. And I think Arrowhead's another good example of this sort of therapeutic. And so it's interesting because you see across these companies, and again, we'll do a Q&A, and I'm going to be eager to ask. This is a warning to the panel members, but just kind of how we're thinking about FSHD drug development and what excites us or what we're particularly interested in. But I think, you start to think about the treatments. So what is going to turn off DUX4 in the muscle to allow it to recover and hopefully improve, versus what is the vehicle that helps get it to the muscle, right? And so there's just a lot of development going on across all these, and a lot of questions, and then as we get more results, we obtain more answers. And so the AeroDUX4 study, so 60 participants receiving this treatment. You'll often see, a requirement for MRI, often muscle biopsy. I think in the earlier discussion, when I look over my career of the last 17 years or so, it is a small minority of patients that participate in clinical research overall. I think there's many reasons that individuals will have want to participate, and that's, absolutely, not only common, it's in fact most people, right? But for those who are interested, I think being very clear about what the trial was, and we're very particular about this because what we look for, maybe this is helpful to speak about, what we look for is unlike clinical care, everybody's entitled to clinical care. Not everybody is entitled to participation in a clinical trial. It's a totally different process. And the reason is, we'd love to include everybody, but in the end, we need an answer. You don't want to run a big trial and then at the end you have no answer. You don't know if you're actually assessed it correctly. So these are very rigorous processes. So what we are looking for is, of course, there's criteria that need to be met. And the reason we have those criteria is that the placebo and treatment groups look as much alike as possible. So that's part of it. Of course, that creates problems later for access, and we certainly recognize that. Importantly, it's not the clinicians who do the trials at the center that decide what these criteria are at the beginning with, but that's an important part of it, right? So And so this trial is currently enrolling. And then just the last one, I'll speak quickly about it. Again, I hope this just illustrates how different these approaches are, right? And ultimately, we need one to be very effective and become available, right? And so ideally, we have multiple, and I hope that we will see that as and I think we will, because that has been the experience in our other diseases where we typically have more than one treatment within a few years of each other. They sort of come on, and then we actually have patients switching between treatments and trying different things, which is even better than only having one treatment. But of course, that first treatment is the biggest hurdle, right? So this is an interesting treatment. So the D4Z4 repeat in the healthy individual is methylated. So there's a little tag on the DNA that tells the machinery of the cell not to express that protein. And so, of course, in FSHD, there's a shortened D4Z4, and then the methylation is absent. And so their treatment goes in and just remethylates that DNA. So again, completely different mechanism of action. And of course, the hope is that this lasts a long time, that this isn't, this could be a lifelong kind of impact, although until we study this, we just don't know exactly. And so this is a very small number, phase 1, as you can see, 6 to 9 patients total. And it's just getting started. And so, and then there's more companies. We're gonna hear from a couple of other companies. And so yeah, so just to sort of recap that, you know, this is a journey for the whole community to go on. And you know, our job is always trying to advocate and balance you know, the conduct of these clinical trials and making sure we do them well. So the other type of individuals we're looking for clinical trials are those who are really committed to doing the trial, have a good understanding of what it involves. But because we hope that they will stick through the whole process, right? As opposed to try it, didn't work, I'll stop. Because again, if that happens too many times, the whole trial is defunct, right? And so I think, you know, that's an important thing to recognize as well. And so yes, I won't belabor that. And again, you know, I think I'll just stop there. I probably have time roughly. But again, if you want to register for our Canadian Neuromuscular Disease Registry, we do put out notifications for clinical trials based on which clinics you attend, et cetera. You have the number there as well. You can give us a call. Importantly for the CNDR, there's no extra visits or anything. You have to just register, and then we collect the data at your clinic. And so it's not like there's special visits or anything. It's just collecting the routine data from your medical visits. Again, I encourage you to explore Move and Move Plus if possible to register. These are as opposed to the CNDR, which is kind of like regular clinical visits. This is more structured, and of course, in many ways, is can be much more useful in terms of that longer term data collection for in a deeper way for individual. And yeah, I'll stop there.
Other questions? That was a lot. Yes. All the trials have like a clinical severity score, or is there a resheet score? So what is that? They all seem to be between the same numbers.
Yeah, so again, you know, we want these treatments to be accessible to everyone. But the ideal group of patients are a group of patients that would change over the time of the study. Meaning if your placebo group doesn't progress, it's very hard to show a treatment effect if the treatment is going to just slow the disease. Just slow the disease, right? Which is, I get asked sometimes, what are you looking for? Are you looking to slow the disease, cure the disease, improve the disease? And the answer is yes, you know, whatever we can get as a starting point. But so what these scales do, the RISI and others, is sort of exclude often more severe cases and sometimes much milder cases to sort of homogenize the group so that so that the treatment of the placebo group is kept progressing at a similar rate on average. Which is frustrating, because sometimes it's quite arbitrary, you know, you feel, I know in one of our ALS trials, if you walk down the stairs, but if you touch the handrailing once, you can't participate. You know, and so then you have people trying to avoid the handrailing, which is not a good idea, right? And but that's, you know, so there's an arbitrary.
Yeah.
Is that related to the face? You also face the face, because I've got placebo in not a double blind placebo control. The test.
In which one?
Phase 3.
It is.
Phase 3 as well.
Correct.
Oh, okay.
Yeah. Yeah, and Fulcrum's a good example that you were commenting on, right? And so I absolutely,
I was in phase 3.
More questions?
Oh, actually, I have another one. Which one of these will be run out of Calgary?
so what my answer to that always is a bit cautious, because having done this for 17 years, there's been many trials where I was in a room like this and said, we've got it, it's coming, and then for whatever reason, sometimes they've just not done the trial, et cetera. So I'm always a bit cautious until we open and have that certainty, but we're certainly hoping to have Avidity, you know, at our site, and then additional ones over time.
Speaking of Avidity, we might have Amy online now. Yeah, any more questions for Dr. Korngut? Okay,
Better make a miracle.
Yeah, we gotta make a miracle from that, okay. Please.
All right, if I speak, can you guys hear me?
All right, what we're attempting to do is have our folks join us virtually. And online on Zoom right now, we have Amy Halseth from Avidity Biosciences. She's gonna be sharing the interim results from their Fortitude clinical trial and provide us with some updates ongoing. So we're hoping to get her on the screen.
I'm here. Can you guys hear me? Oh, I see myself.
Oh, there you are.
Very weird.
Just wait, oh, I'm just waiting for, yeah, sharing with Amy,
Oh, and poor Amy can probably see herself
but
on our screen.
let Amy
Can you
just,
hear me, Amy?
Yes, I
Okay.
can hear you. Can you hear me?
the volume on your
Kind of.
yeah, I will.
We just need to try.
Let me just, yes.
No.
Wait, there, okay.
But I think this will help.
The microphone closer to the speaker, in February at the meeting, I don't know if that.
We're plugged in, let's see if it.
Let's just have Amy say something.
Hello, I'm here. Can you guys hear me?
Yes, awesome. We can hear you loud and clear.
Awesome. All right. And now I'm not sure what you guys are seeing. I'm trying to share, but for some reason my computer does not want to. Oh, here we go.
We can see you and we can see, there we go. Now we have presenter mode. We're good.
Oh my God, it's a miracle.
Thank God.
It took less than 3 minutes and we seem to be working, so that's great.
Sweat dripping off.
I know, I'm feeling a little palm sweaty too just from that. All right, so should I get started?
Yes,
Yep.
please.
Okay. Well, first of all, I'm sorry I'm not able to be there in person with you all today. I had an event happening here in San Diego where I live and where Avidity is based this morning that I needed to be in town for. But I'm really thankful for the organizers for inviting us and also for giving us this virtual option so that we still can present. I'm sad that I'm not able to meet more of you and see some of you that I already know in person. But hopefully this will still be a great interaction and we can, I'm happy to go through this to share. And so what I'm going to be talking about are some early results from Fortitude, which Dr. Korngut skipped over, thank you. But I understand there's going to be a whole talk on it, so of course he didn't want to go into it. But we'll go through that. And the first slide that I'm going to show, just to put it up, that we're saying might change, all this to be aware of. And so from the legal perspective, of course, it's good to say that, but it's also true that things are changing all the time. And we are doing additional studies, we're making new decisions, we're generating more data. And what I'm showing you is from a point in time. And unfortunately, the data that I'm able to show you today is a little bit dated. It's from last year. And when we get to the trial design and you look at how long the various things take in the trial, you can kind of do some math and realize that we're probably very close to having new data, which I can't tell you for sure that that's true, except that I will tell you that we will be having new data coming out some later this year. But unfortunately, I'm still going to be telling you the old data. So I'll try and make it a little bit broader and be a little bit more focused on clinical trials in general too. But also I would say just watch this space for Avidity as things are continuing to evolve. Okay, so I mentioned Avidity is based in San Diego, and we are a small but rapidly growing company that really focuses on muscle diseases, and in particular muscular dystrophies. We have a program in phase 3 for myotonic dystrophy, another program that's about to enter phase 3 in Duchenne's muscular dystrophy, and then our program for FSHD. And you see that we have this AOC, and that stands for antibody oligonucleotide conjugate. And I'm going to share with you what that actually means and then how we think the drug works. So you may see AOC 1020 on slides. We actually now have a generic name, which we abbreviate Delbrax. So you'll see that on some slides as well. So what is Delbrax? So as I mentioned, AOC, antibody oligonucleotide conjugate. So the antibody is this big blue circle thing. I don't know what you call that. This little 3-part thing. And an antibody is just a protein that has a very high affinity to another protein. And it has an oligo, which is conjugated to that antibody. In our case, the antibody binds to a protein that's highly expressed on muscle cells called the TFR1 receptor. And the only reason that we care about this part of it is what that does is it brings the oligo inside the cell. So the oligo is the piece that's really doing what we want it to do in terms of treating FSHD. And the way that it works for treating FSHD is that this oligo binds to any DUX4 mRNA that gets degraded. You therefore don't make the bad guy DUX4 protein, and then you also don't make all of those bad downstream effects. So the idea being that you're going to decrease the amount of accumulation of decreased toxic proteins, hopefully change the muscle into sort of a healthier state, and then we'll have to learn to see, well, what does that mean in terms of function? What does that mean for disease progression? But, you know, given the strong data really around DUX4 being the bad guy in FSHD, we are directly targeting DUX4 in the cell to hopefully prevent all of that bad downstream stuff from happening. And so the trial I'm going to share with you some information on today is our Fortitude trial, and this is a phase 1/2 trial. And so that means that this trial is really doing a lot of things. It's phase 1, meaning it's the first in human study. And so this is the first time that this compound went into people at all. And so the first thing you need to do is be really careful about the safety, right? You don't want to give somebody something new. It's never been in humans. You don't want to treat 100 people at once and then, oh, shoot, now we've learned something really bad about this. So you start small, you start on low doses, and you go relatively slow at the beginning. And so that is what we did here with our first cohort, which we called cohort A, where we started with a low dose. We actually started our first dose at 1 milligram per kilogram, and that's the amount of the oligonucleotide given intravenously. 6 weeks later, you would get another dose and a third dose, and then every 13 weeks is how long, how frequent we ended up giving these infusions. And after that first one at 1 milligram per kilogram, we checked for safety. If it looked okay, we moved on. And so then after that, it was a 2 milligram per kilogram cohort. After we got to a certain point in this cohort A, we were able to take a look at the cumulative safety, realize that it seemed safe. We didn't see any issues that were arising. And so then we went on with another cohort where we went to a higher dose, which is this 4 milligram per kilogram. And then using that same dosing schedule, we had more people in this cohort to really try and understand still the safety and tolerability of the medication. But also what we were able to do in this trial is really start looking at some of the exploratory efficacy endpoints as well. So in both of these, we had both placebo-treated people and people receiving Delbrax. This whole thing lasted for 1 year, and then patients were able to roll into what we call an open label extension study, where everybody, whether you initially got the Delbrax or placebo, everybody would roll on to the Delbrax. And so just when you think about, well, how does this work? What's the trial? So we enrolled people from 18 to 65 years old. We did have muscle biopsies that we performed as well, so we could look at the amount of drug that's getting into muscle and try and answer some questions there. And then we had some of these exploratory efficacy measures as well. And so then in terms of who gets into the study, and I appreciated Dr. Korngut really talking about trying to have a somewhat homogeneous population, because, I'm sure just even looking around the room, I can't see you all, but we recognize how heterogeneous and variable FSHD can be. And when you think about trying to understand if a whole group of people is showing progression or not showing progression, when people are starting way across the whole range of scales, it's actually very hard to do. And it really makes your data interpretation quite challenging. So that's why we have these inclusion criteria, where we're trying to get a somewhat narrower population just to try and answer the question, with the idea then that we can hopefully generalize that in terms of who's eligible to receive the drug. But in terms of who actually made it into our trial, you can see we had more men than women, on average in their early 50s. So as I would say, very young or in the prime of their life. We were open to having people with either FSHD 1 or 2, but all of the people, and this is just in cohort A, had FSHD 1. That's actually true for cohort B too as well. That's the one little preview I can tell you. Just given how much less frequent FSHD 2 is in the population, we have not seen that in the early parts of our trial here. In terms of an FSHD clinical score, I know that was a question earlier. This is a scale that goes from 0 to 15, and on average, people were around 9. But when you look at the standard deviation, we really had people ranging from 3 up to about 14. So even though we try and have these criteria to make it somewhat more narrow, it still is quite wide. And if you think about how long people had symptoms of FSHD, now this isn't necessarily diagnosis, this is asking, well, when did you first notice symptoms? People on average reported sort of in their mid to late 20s. But again, this standard deviation means that we have a very wide range of when people first experienced symptoms. So we had patients who said, at 2 years old, I first experienced symptoms. We had some who had only been experiencing symptoms for a couple of years. And then just looking at quantitative muscle testing, this is what we call percent predicted normal. And so what you do for this is you measure muscle strength in a number of muscles across the body, you put it into some equations, and then it's able to calculate predicted normal based on age, sex, and weight. And so on average, our population had about a third the amount of strength you would expect for people who are the same age and sex and weight. So as I mentioned, this was a first in human trial. So the first thing we always want to make sure about is this drug safe and is it well tolerated? And so we talk about adverse events or side effects. And with all of those, the way this is done in clinical trials, you always go ahead and look at mild, moderate, and severe. And then you look at the specific types of events that are reported. And then there's another kind that's even worse than severe, which is called serious. And so those are the ones that would lead to somebody being in the hospital or in a life-threatening situation. So yes, across both the 2 and 4 milligram per kilogram groups, you do see adverse events. And some are thought to be related to the study drug based on the investigator's opinion. But what's really nice from the true safety perspective is that we did not have any severe or serious adverse events in this. Nobody had an adverse event that said, oh, I think I need to withdraw from the study, and no adverse events leading to death. And in fact, nobody from either cohort A or cohort B, those first 2 cohorts, withdrew from the study. So when you think about the ones that aren't necessarily health threatening or super risky, but kind of the more common things, what did we see? And so we look at the ones that are considered to be related, that happen in 2 or more people. And what we saw is fatigue. We saw a couple people with rash, some hemoglobin decreased and anemia, and then chills. And as I will say, as we've continued to gather more data, fatigue has remained one that we see, although we do see that in both placebo and Delbrax treated people, as well as hemoglobin decreased. Okay, so the first question, right, I walked you through earlier how Delbrax is supposed to work and what it's supposed to do. And this first step is binding to this antibody and getting into the cell. So this is the first time it's been in humans. We actually need to measure that. And so this is just showing the concentration of the oligo inside the cell. I know it's not really compared to anything, so how is this a good number? Is this a bad number? But what I will say is we were very happy to see this amount of drug getting into cells. It's similar to what we've seen in animal muscle, where we know that we have effects. So as a first step of, step one, drug needs to get into muscle. We observed it. We're very happy with that. And that leads us to move on to then step two, right? Okay, so you get the drug into muscle. The next thing you want to know is, are you actually leading to a decrease in DUX4 mRNA or protein? Now, this is actually very hard to measure. So we don't measure DUX4 directly as it's expressed at very low levels. So what we measure are downstream genes, things that when DUX4 is turned on, it turns on other things. And so we're looking at the step one down from DUX4, and we're looking at this in the muscle biopsies. And we were very happy to see reductions in these DUX4 regulated genes, we call them, whether we used a panel of 4 genes that we decided were a really good sort of overview of what to look at. This panel here is a panel of 6 genes that was used in the Fulcrum trials, and then we had another group of 41 genes from the literature. And so what's nice is that these are measured using slightly different technologies and different pools of genes. And so to be able to show a pretty consistent result here in terms of a consistent knockdown gives us a lot of confidence that now step 2 is also happening the way it's supposed to. So we're getting the drug into the muscle, and then once the drug is in the muscle, it's doing what we would predict it to do, which is to reduce the DUX4 expression. But these are from muscle biopsies, and muscle biopsies are not something you can do, could repeatedly or regularly if you want to study a different dose or a different dose regimen or something like that. There's a lot of challenges with muscle biopsy data, and actually the data is extremely variable as well. You have to try and biopsy very close to where your previous one was, and there's a lot of challenges with that when you're thinking of trying to target a 3D object and getting a very tiny little piece of muscle. So another thing that we've been working on is looking at something that we can measure in the plasma instead of from a muscle biopsy where we just need a blood draw, and looking for things that are regulated by DUX4 and do those change in plasma. And in fact, we have identified one, some work with a collaborator at University of Washington, where we are able to demonstrate one of the known target genes of DUX4 does actually circulate in plasma. And when we give Delbrax, we're able to see in the people who receive Delbrax a reduction in this compared to the people who received placebo. This was very preliminary data. As I mentioned, this is about a year old. We have a lot more information on this. And like I said, pay attention to what's happening with Avidity. Later this year, we'll be sharing a lot more data on this. But I think it's a super exciting thing, both for our own program and maybe some of the opportunities it gives us in the regulatory space of perhaps getting our drug approved sooner, as well as things like understanding different populations or different dose regimens or things like that. But I think this could potentially be very important for the field as well. So fingers crossed on that. We also measured and looked at creatine kinase, which is a protein that leaks out of damaged muscle. And another example here where the people who receive Delbrax are seeing a reduction of about 30% in circulating creatine kinase from baseline. So we believe this means that the muscle is in general healthier, but more work ongoing here too. But again, these are both very promising signs. So then the next question is, what about anything related to efficacy? And so again, this is very early data, and this is just from those first 12 people in cohort A. But we were able to see, even with this early time point and early small sample size, is trends where we're seeing reductions or kind of no change in the placebo group, but in general, you're seeing things declining in the patients who are receiving placebo, whereas you're seeing sort of a stabilization or slight improvements in muscle strength when you're looking at the people receiving Delbrax. And again, this is, I'm not actually sure that I said that, this is only from the 4-month time point. And again, we have much longer term data now. And again, I'll say it one more time, that we'll be able to share with the community later this year. And then again, similarly, another measure we did was reachable workspace, which focuses on the kind of the strength and the mobility that you get of your arms moving through these different quadrants. And what you basically want to see are higher scores in this number, meaning that you have more area that you're able to move your arm around in as you get on the improving side. If you're moving in this declining side, when you compare from baseline to 4 months later, the people in the placebo group here were getting worse in what they were able to do in terms of reachable workspace versus improving or maintaining on the Delbrax people. And there's different quadrants here which relate to sort of being up above your head or down below, or even reaching behind you. And the nice thing is that we had a pretty good consistency here, whichever of the ways we looked at it, whether it was the 2 arms or just the 1 arm, and which quadrants we looked at, we saw pretty similar trends. So based on all of that, we have a few things that are ongoing here now at Avidity since the last year. Both the A and B groups have completed their 12-month of treatment, and we have fully enrolled and now ongoing what we call cohort C, which is our quote biomarker cohort that's really focused on that circulating biomarker and muscle biopsies. And this is something that in the US, we hope the data from this group, we can work out something with the FDA to try and get accelerated approval. So instead of going for a full phase 3 program, there may be an opportunity to use some of that early data to get the drug approved sooner, which is unfortunately a US only thing, but it's still good. And then in addition to that, though, we have a phase 3 study. Here it's actually shown as part of Fortitude. I will tell you that that isn't quite how we've decided to run things, and instead we have a true phase 3 separate study that we're in the process of getting up and running right now. And our corporate goal is to have this activated in the first half of this year. So, you know, for people who pay attention to things like quarters, you can tell we're getting very close to that. And so we'll be making, sharing more information about that later this year as well. So how do you get involved? How do you learn more? Clinicaltrials.gov has more information about Fortitude and other Avidity trials, but also trials from other sponsors. So if you really are interested in trials, you know, you can go to clinicaltrials.gov and search for FSHD. There's little boxes you can check for currently enrolling or, you know, the different stages. And that's a good way to sort of learn what the landscape looks like, especially as things change. If you want to learn more about what's happening at Avidity, you can go to our website, the FSHD Society, and then the Canadian TRN, sorry, I forget what those letters are, also sound like a great source of information. And then, of course, your doctor will be able to hopefully keep you informed as well. So I think there is a lot coming down the pipe for FSHD, and I think it's a really good time. And if you can get involved, you have the time and you have the interest. I know there's a lot of ways to get involved in, whether it's a drug trial or somebody who's doing a questionnaire or surveys or other things to maybe validate endpoints. All of those are super important, and all of us would be very grateful for anything that you can do to help us move along. So I just want to leave with this. This is Avidity. This is actually about a year and a half ago, and we have even grown since then, but it really is a huge number of people who are working on FSHD as well as these other conditions, but really are touched and motivated by all of the patient stories that we hear. So know that you all are, you know, why we do what we do. And I'll just leave it by saying that if you have questions specifically for Avidity and you want to reach out, you can use this patients@aviditybio.com, and a real live human will reply to your emails. And yeah, we would hope to hear from you in the future. So I will wrap up there.
Thank you, Amy.
Thanks. I'll stop sharing.
Yeah, we go with that picture, it had like 12 people in it. I don't know if you remember those days, a long time ago. Couple questions for the panel discussion that Dr. Korngut will facilitate if you're okay with that. We'll be holding the panel after we have a couple more speakers. So thank you, Amy. Appreciate it very much.
Thank you.
All right, next up, we have Dr. Brian Mitchell from Satellos. He's right there with the headphones on. And Satellos is a biotech company that's focused on muscle regeneration. And we heard him speak in Toronto a few weeks back, and they've got some interesting stuff going on. I think you guys would be really interested to hear what Dr. Mitchell has to say. So take it away, Dr. Mitchell.
And can we hear me? Is that all good? Volume's working? Okay,
Yeah, we're good.
perfect. Wonderful. So let me quickly share here. See if I can follow in Amy's footsteps and get this set up appropriately on the first try.
You got it.
Okay, there we go. So I'll echo Amy's comments and apologize for not being there in person. I'm in Toronto, and I would have gotten a lot of trouble as today was Dad Day with the young kids, and so my options were to stay home and make sure I take care of the kids. So glad to join you virtually here today and tell you a little bit about Satellos. This is going to be a slightly different story, but I hope something that you'll find interesting, and I hope something that will be applicable to folks that are dealing with FSHD in the near term. So Satellos we're a small biotech, very small, about 20 folks. We're based out of Toronto. We've got folks in the US as well, and speckled across Canada a little bit. And we are working on developing a small molecule pill, a classic tablet, that is designed to help the body respond to muscle damage and help skeletal muscle heal and repair itself. So I'm going to give you a little bit of a background on that and where we're applying this technology first, and then I'll try to link it to FSHD as often as I can. So Amy, your forward-looking statement slide was a lot more inviting than mine, and so I've taken some notes to change ours in light of yours. So Satellos is developing a drug called 3247 that we believe is based on very disruptive science and represents an incredibly novel approach to treating degenerative muscle diseases. We've designed this pill to be well tolerated, an easy to administer medication, and we want to apply this into multiple areas of enormous unmet need, starting with Duchenne muscular dystrophy, which some of you may be familiar with, and some of you may not. So I'll give you a little bit of background on Duchenne, only to kind of contextualize it for what I hope to be future discussions on FSHD. But Duchenne is a genetic disease. It has a single gene where mutations occur, causing the loss and the expression of a particular protein. And ultimately, that results in skeletal muscle tissue being very, very fragile, and effectively daily living and activities of daily life result in extremely serious muscle damage and tearing. The muscle system is a wonderful organ. It actually has the capacity to repair and regenerate, and so it tries to heal itself. But ultimately in this disease, what we see is year over year, muscle tissue is lost throughout the lives of young men. Ultimately, Duchenne patients will succumb to this disease usually before their third decade. There is no cure, there is no effective treatment. What Satellos is focused on is attempting to enhance this regenerative organ's natural capacity to heal itself in the hopes that it can keep up with this damage. So this is the first connection to FSHD. So Duchenne is a completely different genetic root cause. However, it involves muscle damage. When muscle is damaged, it has the capacity to repair and regenerate itself. In FSHD, we have the DUX4 protein, which, as Amy very smartly put it, is a villain. It's there doing toxic things to your muscle tissue. It's causing muscle tissue to also get damaged and break down. And the muscle system in FSHD muscle should also try to repair and regenerate itself. And so for a number of years now, Satellos has been studying this process of muscle damage and muscle repair in the context of Duchenne, as well as FSHD. And we have designed a pill that effectively helps the muscle to perform this natural action in a much more robust fashion. Now, I'll lose my scientist card if I don't have at least one complex diagram or cartoon. And so here it is on the right. I will try to walk you through this. You don't need to memorize any of this, but I'll use it to tell the story of muscle regeneration. So we have there in red a muscle fiber. So we all know muscle is composed of fibers. These fibers, you can think of them like cables that basically contract or expand to help you use your muscles. When that happens in a very sort of severe way in an otherwise healthy individual, or maybe not in such a severe way in a Duchenne patient or in an FSHD patient, that can cause muscle fibers to tear and rip. And when that happens, there is a particular population of cells that live inside of that network of fibers called muscle stem cells. And their job is to do absolutely nothing unless there is damage like this, or even a signal for muscle growth. If that occurs, muscle stem cells wake up and do one thing and one thing only. They divide and make 2 new cells. They have to make a choice when they divide to make 2 new cells. And that choice is either make 2 new stem cells, both of which are absolutely useless for healing muscle tissue, or they go through a special division that science nerds like myself and stem cell biologists like myself call an asymmetric division or an unequal division. And that is at least what we are attempting to show here in this diagram. So that green teal cell will go into a division. It makes 2 new things. One of them is a green stem cell on the right, makes a copy. That copy now keeps the stem cell pool intact. It goes back down to the muscle fiber and waits for more damage. But the other cell it produces, which we've got on the left in purple, is called a muscle progenitor cell. Now, this is the special cell that absolutely needs to be made when there's muscle damage, because what it does, as you can see there below it, is it goes on and divides over and over and over again. And that is the process where we make new muscle cells that can actually go down to repair this damage. So this process of asymmetric division is fundamentally required to repair and regenerate muscle. And what we know in Duchenne and what we've uncovered in additional other degenerative diseases is that this process of undergoing asymmetric division is perturbed in some way or impaired. It's not happening as often as it should. And so we've developed a molecule that basically interacts with some of the decision-making machinery in the stem cells and convinces them when they're dividing, please don't just make useless copies of yourself. Instead, go down this asymmetric division path and make progenitors, because we know that they will go on and do a very good job of repairing muscle. So that's about the simplest way that I can describe this mechanism. We don't need to go through the way the drug actually works or what it interacts with, but effectively this pill is designed to enhance the natural process for repairing muscle. So we have done a lot of work in different models of Duchenne in mice. We've done multiple other models in other muscular dystrophies where we know there's serious muscle damage. And every time we treat mice or even dog models of these diseases, what we see in a very short period of time, a matter of weeks, if not month over month, is the muscle repairing itself and becoming stronger. And it becomes stronger quite quickly, because that's what happens when you actually repair and regenerate damaged muscle fibers. They can do more work. So we have published a lot of information at different conferences around the world over the last 7 years, demonstrating this very convincingly in animals. And most recently, we have now moved this molecule into the clinic. And so I'm going to quickly go over the small amount of clinical work that we have done to date and describe a little bit of the preliminary and exploratory, not controlled and not statistically significant efficacy that we believe we're seeing emerging from some Duchenne patients. So we have performed the sort of requisite phase 1 healthy volunteer study that every brand new small molecule has to go through. We treated 72 individuals across what are called single ascending dose and multiple ascending dose cohorts. We saw a very, very, very mild profile, extremely limited side effects came out of this study. Everything was mild in nature, and only at the doses that were above our predicted efficacious dose of 60 milligrams per day. So we were very, very happy with this profile. What's not on this slide is that we also generated the PK profile or pharmacokinetic profile. Basically, we saw the drug hanging out in the bloodstream for the amount of time that we wanted and at the levels that we wanted in comparison to everything we've seen from all of our animal modeling, where we saw a nice, strong efficacy effect. So this trial was very successful. Excitingly, the second arm of this study was in Duchenne adult patients. So we had 72 healthy volunteers, and then we also treated 5 Duchenne adult patients. Now, all of these individuals were in their 20s. Duchenne patients in their 20s are almost always non-ambulatory, in a wheelchair, limited upper body function, with quite advanced disease. The goal for this study for Satellos was to confirm safety in PK, as well as to explore whether we could see any preliminary signs of this molecule having an effect on any sort of functional readouts or serum-based readouts of calming down the damage in these individuals. We weren't expecting to see anything. We really just wanted to get that safety in PK, but this is a 28-day study, very short period of time. However, once again, we knew from all of our preclinical work, and we know from the biology of muscle, that muscle regeneration takes about a month for a cycle to go forward. So it was theoretically possible to see something, although we certainly weren't hanging our hat on that notion, nor did we have this study powered to show a statistically significant effect. The key take home was that the molecule in patients, just like healthy volunteers, was certainly safe and well tolerated. No new findings in the Duchenne patients. Each of these patients was on steroids, either prednisone or deflazacort. That's standard of care in Duchenne. And so we confirmed that our drug did not behave any differently from the sort of PK profile in the background of steroids. And what we were really excited about is that we think we may have seen a trend in improved grip strength. So grip strength was one of the functional measures that we looked at in these individuals. Each of the individuals in the trial were actually able to perform this test. We did not have any entry criteria that required individuals to be performed. We really were just trying to learn anything that we could. So people did have different levels of strength coming into the study, but we still managed to see about a doubling in the average strength between before and after. And in terms of natural variance in this group, that is entirely unexpected. So Duchenne patients in their 20s, and especially with the numbers they put up at baseline strength, this is certainly outside of regular variation and well beyond the sort of retest variation of the instrument. It was driven by 3 out of the 5 patients. They had these increases in both of their hands, and the other 2 patients were actually stable over the 28 days, no real changes. We looked at pulmonary capacity and again, some serum markers of inflammation, and also put on a device that looked at upper body effort, and all stable across the board there, which was basically the expectation, but we just didn't know. So we wanted to take a check and look. So each of these patients will be invited back for an 11-month follow-up later this year, where we'll continue to assess these things and hopefully see these changes build. That's exactly what we see in all of our preclinical work, month over month over month. Everything always increases and gets better, as one would expect from further and further cycles of regeneration. We're also going to be giving each subject that returns the option to participate in an MRI, so hopefully we can capture some additional objective measures of things changing in their upper limb muscles. So beyond that phase 1, we are looking to get a phase 2 in pediatric Duchenne patients up and running later this year, and this will truly be an efficacy study, placebo-controlled, randomized controlled trial, where we will ultimately try to look for objective effects, both through biopsy and MRI, as well as a whole slew of muscle function testing on the upper and lower body. These will be ambulatory individuals in Duchenne. So I realize, I'm not giving you information here on FSHD trials and when they are going to start. Satellos has a goal of trying to demonstrate effect in Duchenne first. This is the indication where we started this work. A lot of the scientific discovery comes from learnings about the gene involved in Duchenne. But we're really excited to apply this outside of Duchenne if we can get the signals that convince us that the drug is translating from preclinical to clinical. If we can truly start to see some impactful changes in Duchenne, our team is sort of ready and chomping at the bit to really start thinking about what's the next indication and where are we going. And we have been talking to Neil at FSHD Canada and SOLVE-FSHD for quite some time about, how can we apply this technology either, in combination in the future or as a standalone in FSHD. But all of that will come in the event that we can really get some concrete data to suggest that this is working as we think it is. And then we'll certainly be in touch with the FSHD community again to talk about some more concrete plans for potential trials in the future. So this is a bit of a public service announcement. Please stay tuned, see what happens with these trials coming up in the future. And then we really do hope to open this up to a wider range of indications with FSHD, I can tell you, at the top of the list. So thank you very much for your time, and I will stay on the line and await any questions in the follow-up.
Awesome. Thank you.
Thanks.
Exciting stuff. We love drugs. Okay, so I think I need to disconnect.
No.
that.
No, I think they want to be able to stay on,
They
right?
can stay on, but they just will be on the screen.
Okay,
Yeah,
sure.
but I'm going, I'm gonna turn this light back on. I know if
Make that work.
Yeah, all right, so,
Here, Beth, just plugging it over there.
you're up, right? Where are you? Oh, there you go. Yeah. You're more ready. Okay, so it's still. You hear it? You're right, my.
It's my stomach.
Yeah,
They sometimes have like big events outside, there could be.
Yeah.
They're like a road for them. So, this is yeah.
So while Beth gets things set up here, are you gonna say something?
I'm gonna say something about you. Can I say about you? All right. This is Greg. Everyone think, oh, Greg. No. Greg Love is the CEO and co-founder of Histone Therapeutics. It's a biotech company at the forefront of precision epigenetic reprogramming. Dr. Black holds a PhD in molecular biology from Tulane and completed a fellowship at the University of Washington where he studied FSHD. He's going to speak to us today about genomic medicine for FSHD, but also hopefully a little bit about Histone, maybe? Okay, right on. Now.
Yeah, I've spoken a lot to the FSHD patient community. I'm a little nervous to give a company pitch about it, so I wanted to preface it with a little bit about genomic medicines, what it's all about. I thought what better way to start talk about genetic medicine than with a sing-along. But I'm not doing it if you don't sing along. So, are you in?
Sure.
Okay, so this is a song I wrote for my children, and it goes like this. You're gonna repeat after me, okay? It goes like this. DNA makes RNA, makes protein.
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 AGCCCCCTTCCCK.
ADTCCCK.
That's the gene that says, I love you. So there's a lesson there because DNA makes RNA, makes protein. And you've heard a lot already about medicines, which are based on DNA or RNA or nucleic acids. And that's what genetic medicine is at its basis. It's a drug that instead of being a small molecule, is a piece of DNA, is a piece of RNA, is a small piece of RNA, is a big piece of RNA, it goes all over the place. And there are many approved
safe therapeutics based on nucleic acids. But what's happening in our communal conscience is there is becoming a blurred line between what is a genetic medicine and what is gene therapy. And they're completely different. They have a different regulatory path, they have a different safety profile, they have a different mechanism. I was talking to Priscilla earlier, and she mentioned reading stuff in the news about how they make dire wolves and woolly mammoths using gene editing, and like, oh my God, are we becoming humans like that? Like, no, right? Yet. So genetic medicine at its basis is not necessarily gene therapy. The COVID vaccine, for example, dare I get controversial, was an mRNA. DNA makes an mRNA, makes protein. The RNA made the protein, which was what your immune reaction was set against. That's a nucleic acid therapy. That's a genetic medicine. The siRNAs that are piggybacking on the antibodies that you just saw from Avidity, the drug is the small RNA. That is a genetic medicine. CRISPR, which you've probably read about in the news, which was used to make dire wolves, is editing DNA. It's going into the DNA sequence and cutting it. That makes it a gene therapy. If you're replacing a missing gene by putting in DNA and then repairing that gene, that is gene editing. One thing you've got to remember about DNA is it is a remarkable piece of chemistry. It is super high fidelity. Think about it. You started with one cell in your body. You have 3 billion base pairs of DNA and trillions of cells in your body. Your cell faithfully replicates that DNA trillions of times throughout generations to create the entire human population. That's a pretty high fidelity thing. So cutting it, pasting it, obviously has its ethical and safety issues that we need to think about. Gene therapy, because you're going in and cutting this high fidelity piece of DNA, is permanent. It's very hard to remove. You would have to go in and redo it. So it's really a one-way street. And it's often done using viruses, which add sort of a layer of complexity to a medicine, depending on what it is that you're trying to treat. And so I think it's important as a society that we think about these medicines in two categories. One is genetic medicine and the other is gene therapy. One being sort of something that wears off after a short amount of time, that is using a nucleic acid basis, but for the most part is inducing something that is not living. Whereas gene therapy is something that is inducing something permanent. Now, what's happening in the world and in the narrative, and we really need to be careful about this as scientists, especially in a day and age when science is literally under attack. We need to have respect for our critics and really walk them through what's happening. And one thing that I've noticed a lot lately is a ton of press around this patient. Very beautiful baby, had a disease called that caused by a mutation in CPS1. It's the CPS1 deficiency. And the baby received a custom CRISPR therapeutic made by Aldefron, organized through the nonprofits, an incredible lift. And they were able to deliver a CRISPR using an mRNA to deliver CRISPR to the liver of this baby so that they could make the enzyme that was missing. It was an mRNA, same as the COVID vaccine, used a lipid nanoparticle, same as the COVID vaccine. But is it a genetic medicine or is it gene therapy? Obviously, it's a gene therapy. Obviously, we've used the same safe delivery technology to deliver the CRISPR, which then goes and cuts the DNA, making a permanent change, and enough of the cells are made in the patient's liver so that they can reduce their requirement on exogenous enzymes by 50%. It's amazing. I'm going to talk a little about, with that sort of philosophy based, I'm going to talk a little bit about this company that we started called Histone Therapeutics. And we talk a lot about the power of the epigenome. So if your DNA is so high fidelity, what is your epigenome? Your epigenome is quite simply put, the way your DNA is programmed. My fingertip and my eyeballs have the exact same DNA sequence. They derived from that single cell when I was a little baby embryo. But they do fundamentally different things. Otherwise, I would see out my fingertip. And the reason that is because of the chemicals that are put on DNA. And what's interesting about this is that means that your DNA code, the high fidelity process that gave rise to it, does not necessarily determine your fate. DNA is organized. It's carefully organized, in fact. It forms itself into these things called nucleosomes, which I've tried to get AI to draw properly here. This took a couple of improvements in ChatGPT. But essentially what I'm showing on the top there is you've got some chemicals that sit on the basis of DNA, and they keep DNA silent. We're familiar with this field because we always talk about the methylation of T4Z4. We talk about the silencing of DUX4. That is literally methyl marks, little pieces of carbon getting stuck onto the gene to keep it silenced. If you're missing those methyl marks, or if there's other types of marks, you get activity. And so that in itself is the basis for the genetic code, for the programs that we run in the software in each of our different cell types. And it's the reason why we sort of, dare I say, die. You know, we might be beholden to genetic mutations, but we will all succumb to changes in our epigenome. They change during disease, they change during exercise, they changed after I watched the friggin' second episode of The Last of Us, which traumatized me last night. And so, you know, but what's really interesting, and we know this in FSHD in particular, is that there are other genes that we know if they're expressed differently will affect the disease phenotype. So there's, just because you have a D4Z4 truncation does not necessarily mean you're going to have severe FSHD. And so that's what we think a lot about histone. That is the power of the epigenome. But it's very important to think about, it's not just the chemical marks near the DNA. Because if you think about it, those methyl marks that you're putting on D4Z4, those are permanent. It's not changing the DNA, but it is giving the DNA a tattoo. And if you fail to maintain that tattoo, that's what FSHD is. So at Histone, what we're focused on, which is kind of a bit of a backwards way of thinking about the world, is we're focused not on changing the chemicals on DNA, but we're focused on changing the chemicals on the proteins in the nucleosome that's around DNA. It doesn't really look like that. Those are just like there for illustrational purposes only. But essentially what we're doing is we have a histone modulator protein that we've designed. We send it with a little protein that's like an Uber driver. It's a nuclease dead CRISPR, much like the one being used by CRISPR biotechnologies. It goes to a specific site of DNA, and then it starts mucking around with the histone proteins. So it's not touching DNA at all. And also importantly, it is not introducing any sort of enzymatic activity into a cell. So it is transient, it is not permanent, it is potent. So my little talking points here is that we're able to change gene expression in a cell without altering the genetic code at all, without giving the DNA a tattoo. We do not use viruses, and we have shown now we can get tissue-specific potential. I'm going to show you that. It's the coolest thing I've ever seen in my life. And of course, we can start thinking beyond the genetic mutations. We can start thinking about how to reprogram. FSHD fundamentally is a disease of misprogramming. DUX4 turns on in a skeletal muscle at very low levels and starts to reprogram it. And so that is what development is. That is what cells do. And so why don't we just do that to make things better? This is the philosophy of Histone. So we coined this little term, which I find super cute. Some people laugh at me, but we call our platform the on platform because we turn genes on. And our goal is to make therapy for genes, not gene therapy. And so if you look at all the other sort of companies and efforts that are driving towards the clinic in this space, there's different attributes associated with them. And my argument here, which I think is very important that we consider as a community and as a society, is that if you meet any of these criteria that are shown in red or green, you are de facto a gene therapy, whether or not you like it or not. And the reason is because the genetic medicine is causing a permanent alteration. And so, for example, some folks have introduced similar types of molecules into muscle cells using an AAV. An AAV is a type of virus. It's used in the approved Duchenne drug, which delivers microdystrophin to skeletal muscle. And that AAV has a little piece of DNA in it, and it gets inside the muscle, and that'll stay inside the muscle for like 7 years. So, you know, we talk about Lawrence's clinical trial map and how you have like a, you know, maybe a 2 to 5 month window to run a well-designed study. You know, it's very hard for a biotech company to run a safety study for that long and really understand the consequences of your gene therapy. So that's why I think it's very important to delineate, once again, the difference between a gene therapy and a genetic medicine. And so what we've tried to do is we've tried to design a modulator protein that essentially goes into cells, does not cause any permanent change. We designed it to be ultra potent so that when it goes into cells to turn on a gene, it does so at very low levels. We are now showing that we can routinely deliver about 20-fold lower doses to get a result than if I was using a normal CRISPR. The CRISPR is there for a very short amount of time. CRISPR is a foreign protein. We do not want it lingering around in muscles for years. We do not have any RNAs, which is this is a scientific nuance, but if you're delivering little nucleic acid via DNA, that DNA gets made in your cells, DNA makes RNA. DNA making RNA is an imperfect process, and if there is a mutation in that over the course of many years, that's a bad thing and very difficult to manage. And we avoid viral vectors, and we avoid any error-prone enzymes. So from a scientific basis, what we're trying to do is create the world's simplest gene therapy. It works. This was a tearjerker moment at my company. And you'll see when I show you my team slide, we saw Avidity's Army, we saw, you know, Ryan's 25 small company, I got a small company. And so when we saw this, we were extremely happy. We delivered an mRNA encoding our molecule in a standard run-of-the-mill lipid nanoparticle. This is a lipid nanoparticle that honestly shouldn't have even worked. Lipid nanoparticles are notorious for draining directly to the liver. And of course, we would have to be very concerned that we were turning on our gene for muscle in the liver. And what would that do? So we just did the experiment. We injected our formulation. You can set, can't point. Okay, so this is a control mouse, right? We don't get upregulated. We're targeting a gene here. It's a therapeutic gene. Companies have to be careful with what I say, but it's a therapeutic gene. If we introduce our construct, we turn on that gene into muscle. You can see it going on from 24 hours to 72 hours. That is awesome. That shouldn't have been on. That was like even to get it into muscle with a lipid nanoparticle of this poor quality was a kick-ass moment for us. But check this out. This is where we detect the stuff that's drained to the liver. So we know that it's going directly to the liver. But when we look at that therapeutic gene that we're targeting in the skeletal muscle, it does not activate in the liver. Now, why is that? The reason that is because we target histones. If I targeted DNA directly, you'd be damn sure that thing would turn on the liver. But histones have their own pattern. They have their own thing going on. My fingertip is not my eye. My histones have different things going on in different tissues. We have to dial it in. We have to get it perfect in the cell type that we're looking at in order to even turn it on. So what we're looking at here is an mRNA that is likely to outperform. If I were to introduce this gene just with an mRNA, I wouldn't get as much mRNA. So we are able to outperform standard mRNAs. We're able to deliver it using standard, non-specific, non-proprietary, very simple lipid nanoparticles, and we're able to turn on genes. Of course, we're thinking about FSHD with us. With this, we've been funded by Friends of FSH Research through a collaboration with France Speardrop, who many people here know very well. And we've now shown that we can turn on very difficult to turn on genes to silence DUX4 activity in cell culture. We just received another grant to go and try this in animals and continue our animal work. So we're really excited about this. But you remember that picture Lauren showed you where we saw kind of my company in the very beginning, where it's like we're talking about epigenetic repression of DUX4. Because we're thinking about turning things on, our world is much bigger than that. We've now shown we can activate metabolic genes in the liver that impact muscle growth. We can turn on genes that impact muscle growth in the muscle. We've built an AI system that lets us iterate through pretty much every single gene in the human genome to try and figure out which genes we can turn on in order to intervene with the mechanism of action that we're interested in. And the world is looking very bright for us. So we're really excited about this sort of very promising technology at its most early stages. And we're going to continue to try, build, and grow this company. As I was telling Debbie in the audience, it's been one of the hardest things I've ever done. It's like working at a university, except it's like you have to build a university, and you have to hire the lawyers, and you have to do the lab work, and you have to be the HR. It's never. We're a very small team. We got a bunch of rock stars in here. So myself and my colleague Alan, we both built companies and you know, taken them all the way to exits. We have Jesse and Nate, full-time FTE in the labs. We have bioinformatics support, both based in Vancouver and Baltimore. But we have advisors around us now that are really helping us drive this forward. Emil was the founding CEO of a company called Blue Rock Therapeutics that brought the phase 3 Parkinson's cell therapy to the clinic. Bruce Wentworth, who many of you know, has helped co-found Dyne Therapeutics, has been working with muscle disease forever and ever. Han Laruda Baker was my co-founder, Shiri's postdoc advisor, married to David Baker, who's an inventor on one of our patents, who just won the Nobel Prize. And James Traeger, a Seattle legend who's brought multiple drugs to clinic, is helping us map out a clinical path. So we are an early stage, but powerful little team doing amazing work. And please, if you have any questions ever about FSHD or what we're thinking about, please never hesitate to reach out. Unlike this guy in Seattle who sells couches and calling them like, always happy to chat. Anyway.
Excellent.
Local scientist has a, oh, sorry. Drug development is not shooting up. But if they're inherently things that you're using non-viruses and other things that would make this more therapy, things that are to it working on this stuff.
You know, affordability is a very difficult thing to think about because, especially from the Canadian perspective, if I save the US hospital system 5 minutes in the hospital, it's like value of hundreds of thousands of dollars. If I do that in the Canadian system, not so much. So in Canada, we really, when these complex therapeutics emerge, we're gonna have to rely on our academic systems to deliver them like we've done with CAR T therapies. This is an mRNA lipid nanoparticle. This is like dollars a dose. And so this is gonna, you know, the way we're envisioning this, if it all works out, is it is literally like, this will be like an Ozempic. It's a redosable, you know, or maybe, but it's a redosable sort of locally accessible medicine that doesn't have complex manufacturing. In fact, the manufacturing, we probably 99% of us in this room have this manufacturing inside of us.
We're going to move on because we will have a time for a panel, but thank you. No.
That's great.
All right, thank you, Dr. Black. Telling me slackers in this room, all these letters are going. We have another slacker.
Dr. Eva Chen, she's the executive director at SOLVE-FSHD. She's a PhD in physiology from the University of Waterloo and completed her postdoc training at the University of Sydney in Australia and at the UT Southwest Med Center in Dallas. She's going to speak to us a bit today about the future of drug development for FSHD and Saul's role in it. Thank you. There we go.
With the arrows, yeah.
Okay, thank you for inviting me to come from downtown Vancouver. Great to see some of you again. For those that I haven't met, pleasure to meet you. So we are an office here in Vancouver. I was recruited to come here, actually living in Denmark at the time, but talking to people like Neil Camarda, running FSHD Canada, and our founder, Chip Wilson, I had my arm twisted to move to Vancouver. So I'm going to talk to you about what we're doing at SOLVE-FSHD, and really we're founded, started in 21. We really launched in March of 2022, so we've been around for 3 years, really to be, a catalyst driving innovation, trying to accelerate getting novel therapies into the clinic. And I'd say our biggest goal is to move quickly and help to build a pipeline. We started with the bold goal of trying to get to a cure by December 31st, 2027. That's a Chipism, Chip Wilson, very goal oriented. Those of us in drug development know that's really difficult. It's probably not going to happen. And I think we've now stepped back and had to patient education and think about, the reality of our goals. So recently we redefined our goals as getting to a solve by December of 2027, which means to get 3 therapeutics into pivotal clinical trials, which are phase 2 and 3. And these would address both the mechanism of the disease, so going after disease modifying therapies, as well as those that can change fat content and or rebuild muscle. So there's a real interest, and there's Chip shown during our launch. He will tell you about the fat that exists and why he would rather have more muscle than fat. And he's coerced by Neil to think the same way. So, but when we launched, we showed this as our strategic plan, and it's been broad. On the far left, we have tried to accelerate some of the critical items that have been talked about here. How do we develop biomarkers which can accelerate decision-making in drug development? Avidity now leading the way with a circulating biomarker. Fulcrum, as was previously discussed before, using MRI as an imaging biomarker. Those are critical for companies to make quick decisions on whether their therapy works or not, so that they'll know whether they should invest more time and money in running the lengthier trials for clinical outcomes. So collectively, we poured more than $6 million into biomarker and other therapeutic grants. Probably it's not well advertised, but we did put almost $3 million into the CTR funding. And that was to support the Move Plus, which added the MRI imaging. So we funded a number of grants, and now we're doing a little bit more internal R&D on biomarkers because we have put money out in the community, but we want to have a little bit more input ourselves. The second pillar there is really to be a catalyst and to make investments in early stage biotech and pharmaceutical company. And I'll talk at the end about the companies that we have put money in, but they're all listed on our website. So collectively, we've made commitments of almost 40 million at this point and trying to get early stage companies going. Again, accelerate more therapies into clinical trials. We're going to overload the clinicians like Leo Wong and Lawrence Korngut. They're going to have a lot of trials in the future. The third pillar is the SOLVE-FSHD solutions. We wanted to start our own harmonized way of doing preclinical research. There's a lot of literature showing irreproducible preclinical research if you fund 10 different labs, so we wanted to harmonize that. Shout out to Jesse Fox, who started working on this with me while he was doing his PhD. He still continues to work with us on this, and I'll speak a little bit more about the preclinical CRO in another slide. And then we always had this idea of a stimulus for innovation, which, Chip was really behind trying to reach out and put out a call to all ends of the earth to get more companies, more teams to think about working on FSHD. And we did that then through an organization called the X Prize. And I'm going to give a quick update on that because we recently gave out a number of awards for X Prize winners. Before I talk a little bit more about our initiatives, I wanted to just reiterate that our focus has been in this red circle. Lawrence nicely outlined the breadth of research. Greg has talked about early preclinical research. So a lot of ideas start with academics, but eventually someone has to test those ideas. Does that therapy work in cells? Does it work in animals? Is it safe? Can you manufacture enough to run clinical trials? So a lot of what we do is to try to drive more ideas from this early identification stage and fund the type of work that generally governments and other investors won't fund. And that's kind of the boring stuff of rerunning animal studies, scaling up your manufacturing, running your safety testing, running toxicology. So we put a fair bit of money into those high-risk steps because we're willing to have, take these higher risk investments. Since the last time I spoke with this group, I guess it was June last year, we have added to our team. Richard Hansen has been with us for a while. He runs our grants program. Karen Lamb had just joined us. She is our head of business development, but she's also an expert in commercializing biomarkers. And then Maya Garani joined us to run SOLVE-FSHD solutions, working very closely with Jesse Fox. So I want to speak briefly about those. So at the end of last year, we again went to our board and said, we really want to take a little bit more of our projects internally. We funded a lot of academics. We think there's been a lot of work done on discovering new biomarkers, but there's been very little done on what you call the validation. The validation is both an analytical validation. Is it reproducible if you run the biomarker test at multiple clinical trial centers or in another centralized lab? And then is it biologically validated? Does it track and predict disease progression? And could it be used in the future for drug approval? And as Amy mentioned earlier from Avidity, we're really looking forward to seeing what their biomarker looks like. And we're happy to partner with any company that has a leading biomarker to help make it available for all companies, or to work with a consortium of others, FSHD Foundation, FSHD Society, FSHD Canada, to try to validate other biomarkers and then make them available to all companies, and also to have more centralized biobanking. Right now, there are cell lines, there are plasma samples, there are FSHD samples in very small secluded biorepositories. Difficult for companies often to access, and we want to make those more centralized. So we're putting some money into that. I want to mention one of the newer investments we made recently was with a company called Transcripta Bio. We just announced this in March. And the main thing I want to show here is that this is a strategic partnership as an example of how to accelerate getting new molecules for new approaches into clinical trials. And I thought I'd try to put together a graphic for you. In this investment, we're now looking at these fatty adipogenic progenitor cells. So I know many of you as patients know that your muscle doesn't regenerate. It tends to become fatty and fibrotic. And while we can work on the DUX4 disease modifying, and we can try to build more muscle, somehow we have to change that shift of cells going from muscle into fat. And this project is specifically focused on that. We're looking at these fatty adipogenic progenitor cells, or FAPs, in healthy versus FSHD patient samples, identifying the misaligned pathways, and then basically screening for FDA-approved drugs that you can then quickly accelerate into clinical trials or yet another approach. So again, as Lawrence went over the clinical trial pathway, we're going to start with some cell-based screening, and then hopefully if we get any hits, because these are FDA-approved drugs, they already have a safety package, we can do minimal preclinical safety and get into clinical trial. And we really hope that that phase from discovery to preclinical to clinical will be about 1.5 years. And that's in collaboration with Transcripta Bio, a company down in the Bay Area. SOLVE-FSHD solutions, I alluded to that before. Again, Jesse's done a lot of work on this, and we feel that the novel aspect that we can provide is the access to some imaging technologies. In this case, we'll just call it an ultrasound plus that we can do in animal models. It's also available to do in patients. The ability to translate imaging from animal models to patients is really important in preclinical research, and currently you can't do that because we don't do MRI in rodents. We have some other, I think, benefits based on real-time reporting. And then on the right, those are just some standard parameters that are provided by many other preclinical CROs. So we wanted to be an FSHD specific preclinical CRO, which we've called Solve Solutions. And we are now testing therapies for a number of companies at that CRO. A few words about XPRIZE. So again, our founder and funder, Chip Wilson, is really a big believer in innovation can come anywhere, can it's hard to come from a garage when you work in biomedical research, but an idea could come from someone's garage. And so he worked endlessly with the organizers of X Prize. So we're both the sponsors of this Health Span Prize, which is about extending healthy aging, where you have to show improvements in muscle, CNS cognition, as well as the immune system. That will be an X Prize where you could win 60 to 80 million. So that's really the carrot that most teams are going after. But we wanted to put FSHD on the map, so we also have a $10 million FSHD bonus prize. And briefly, it is a competition that was launched in 2020, end of 23, early 24. We got a number, there were thousands of teams that entered. We had hundreds that completed the submission. 150 for the Health Span Prize and 40 some for the FSHD bonus prize. So where we are now is we just finished the semifinals. Sorry, we just finished the Milestone 1 Awards where we gave 8 teams $250,000 for their FSHD bonus prize idea. And so we're expanding the number of teams and companies interested in FSHD. There will be a semifinals for the Health Span Prize coming between 25 and 26. We will not have a Milestone 2, but we will encourage teams to continue to enter the FSHD bonus prize. They need to submit clinical data by 2029 for the payout in 2030. So that's a bit of a longer term vision. And then here's just a few pictures from the award ceremony that we recently attended in New York, where we gave out these milestone awards. And if anyone doesn't recognize the 2 gentlemen in the center, there's Chip Wilson and next to him, Chris Carino. So those of you involved in the FSHD community, Chris Carino is known in the US as the voice of FSHD. He's an announcer for the Knicks. And again, this first time we had a chance to meet him and hear his story, and he's done amazing things among many other patient-run foundations. And here's just a quick snapshot of the names of the teams on the right and the types of therapeutic strategies that win the $250,000 bonus prize. So some are genetically driven and others are going again after immune system or DUX4 regulation. I'm going to spend a minute on the slide. So what we have been trying to do, as I mentioned earlier, is to rapidly accelerate therapeutics. I know the FSHD Society has their picture of the pipeline. We keep our own that we track. So as we know, Fulcrum stopped after phase 3. Avidity is now in phase 2, going into phase 3. And there's a number of other companies that are progressing and that are either in clinical trials, recruiting or not recruiting. But this dotted line here represents companies that have filed an IND or a CTA clinical trial application that will soon be recruiting. And those on the left are still in earlier phases of clinical trials, getting close to starting the clinical trials. And so we're trying to, where we can, help support those companies to accelerate them to getting more clinical trials going. Again, in the time frame of trying to get them to pivotal trials by 2027, which is only a few years away now. Finally, because I was asked to speak to the future of drug development, when I started, I outlined that we need disease modifying approaches. So we need to go after the molecular cause of FSHD, either through the epigenetic mechanism or through DUX4 modulation. And there's a lot of different modalities. So we're modality agnostic in terms of the companies we fund. But we also realize now that we're going to need to harness the immune system, the inflammatory system. That's probably part of what regulates those FAPs. And then we know there's much interest in muscle regeneration. And really the future will be a cocktail of therapies. And I think anybody who's had family members go through cancer or cardiovascular disease, the future will be a combination of therapies. And I think we do have to think carefully about how we will develop those. So I just have one slide and I'm happy in the future to speak more, but I've given talks on this, like how do we develop these combo therapies? From a drug development point of view, they're not that easy. You have to actually test them separately and then you have to test them in combination. If you have a drug that is, again, already FDA approved for something else, it's called a repurposed drug, you can go quickly. If you have what's called a new chemical entity or NCE, You have to do your safety studies before you go on clinical trials, but then you could quickly combine it with a drug that's already FDA approved. And you can have all sorts of combinations. These will, again, cost time and money, but I think in the future, we'll find, just like in the SMA community, we'll have small molecule therapies, we'll have genetic medicine and gene therapies, and we'll have to work out which are the best in what order. Because some molecules may be effective quickly. I used to work on muscle modulators that acutely improve muscle function. So you can take those while you're waiting for the gene therapies to have an effect, because those tend to take a little bit longer. And then finally, some of you are familiar with, we do host a meeting up at Worcester every year, and we bring in the top academic clinicians. We bring in many of the top companies from around the world. And we have this meeting and we make sure everybody sits on the stairs nicely together for at least one photo. But I just wanted to share some of those take-home messages. Something that doesn't come up often, although I've had some general conversations here, there's not a lot published, but we did have one person present to say, exercise is beneficial, but it has to be the right type of exercise. It has to be prescriptive. So, and I think we need more consensus information on that. We clearly need to understand the role of what we call non-hygienic cells. We know we want more muscle, but we have to also change and modulate these FAPs and immune cells and inflammatory cells. And eventually, how do we treat those in combination? I think there is emerging data on how ultrasound or other types of non-invasive imaging could be used, that you could do point of care in addition to MRI, as well as working on circulating biomarkers. And then I would like to say, obviously, the work on these natural history studies, the MOVE and the MOVE Plus are really critical. We're also trying to help support Jeff Statlin and the CTR on the pediatric natural history study. And I have calls regularly with the parents that have children with FSHD. And so we're working with them on a retrospective natural history study, because that information can be available sooner. So we're trying to work with all members of the community to accelerate therapies. Again, a lot to be coming in the future. The future is nearby, and I think we will have a combination of effective therapies in the near future. So with that,
All right, you want to stay up here? Okay. All right, yes,
Dr. Korngut, would you like to come and give some really hard questions to these folks? Let's plug in, well, we maybe just 5 minutes, but we're gonna do this too. I think because this is what everyone came for.
So maybe I'll invite Paul.
Yeah.
You know,
Yeah.
and,
We'll put these guys up on the screen.
we have Ryan, Amy.
Yeah.
Online.
Miracles are happening.
Miracles are happening. We're getting you back up on the screen.
Okay.
Look, there they are. Woo! Now, Amy, could you say something? Let's see if we can hear you.
Yes, can you hear me?
Yes. Good.
Great.
And can you hear, how about you say a few words?
Can you hear me? Yes. Amy?
Yes.
Yep.
Yeah, I can hear you.
Yeah, that's great. So maybe just to start off, you know, I'll just ask the audience if there's any questions from anybody that the group is looking to ask. We didn't have sort of a full Q&A after each session, but is there anything that's pressing, or then I have some questions I can do. So.
I actually just have one question for you. The last slide was all of 2025 from Worcester. You're saying the first thing about exercise being beneficial, but there's something coming down the pipe about like what type of exercise? Is there somebody accompanying that?
well, we had somebody present at the Worcester conference, and he looked at the body of literature, and what he said is we don't have an information statement like that. In other fields, you will often have groups. American College of Sports Medicine in particular has done that for other indication. So unfortunately, it's not, I guess the question is how best to translate that information and how do we get that out there. I've hunted back to see if the FSHD Society or any chapters are interested in helping to work on that. We had several exercise physiologists that were there, and that's my background. So I think the information is out there. It just has to be documented in a way to make that accessible to patients. But exercise is medicine, it has to be the right type of exercise for any indication, and so I think we just have to get that, I guess, knowledge translation somehow.
Yeah, this question's actually for you.
Okay,
And we've kind of heard a lot of people talk about it today, that oftentimes in clinical studies, people that are under 17 or 18 or over 65 or are non-ambulatory are excluded for the reasons that you described. And you made a comment that said that can affect access to care. Would you say categorically, if there is a treatment, that those people that were excluded from the trial will be not have access to the treatment?
yeah, so so I can't speak to other countries in as much detail, but certainly in Canada, the process is the results of the trial, if it's positive, go to Health Canada. Health Canada provides a regulatory review. So they don't worry about the price of the drug, it just does it work, does it not work, and who should have access to it. Generally, when they approve, they will approve it for anybody with FSHD. However, then what happens is there's a reimbursement process, Canadian Drug Agency gets involved, they review, and then typically what they say is the drug has only been shown effective in this group of people, which is, let's say, above age 18, below age 65, with these parameters. And then there's a process, so it's not that they just accept everything, but at the end of the day, there will be some elements of that criteria that limits reimbursement. And due to the cost of, modern therapeutics, you essentially don't have access, right, for 99.9% of people. And so then we use our registries, et cetera, to advocate and say, look, there is benefit, and companies can be helpful in certain circumstances. There'll be patients who maybe were 64, but now they're 66, 67. And so we collect real world data and then provide that. And we're actually partnered with Canadian Drug Agency to meet their needs for decision making. But that's the concern. But there's just no way around it. And there was,
I've got a question about it, because you had brought up that, I think it's Phil Chamberlain, right, that's doing the work on the blood biomarkers to replace biopsies.
right.
Is that something that SOLVE would look at, maybe picking up as well, and sort of, helping to support that work? Because, obviously, we only have so much muscle, so the concern about having biopsies for every single drug trial is a concern as a patient, so it seems like a way to move forward.
I think in the context of those are the types of partnerships we'd like to stimulate. It's the work of Avidity, and Amy can speak to that, but we've certainly had conversations. I think our goal would be that where it makes sense, that we help to make it available more broadly, and then it helps build the case for that biomarker. I think we've just had some preliminary discussions with Avidity, but Amy, do you want to add anything in?
Yeah, I didn't hear the entire question, but I think it was basically like, can we stop doing biopsies now because they're terrible?
And how do you make your biomarker available?
Yeah, it's really building the whole evidence around the biomarker right now, because right now it's just kind of an observation of it goes down when you give our DUX4 targeting therapy, but we really need to do a lot more about understanding its levels across people with FSHD. Does it change as you get older, or as your severity changes? All of that work. And so for now, a lot of that does relate to also correlating it with muscle biopsy data and trying to kind of build that story together, that what we're measuring in the circulation does reflect what's happening at the muscle, or actually what we think is better than measuring what's just happening in a teeny piece of muscle. And I'm an optimist. I hope in the next few years that we won't have to rely on doing additional muscle biopsies for this specific thing. There may be other questions that we need muscle biopsies to answer in different areas, but in terms of, are we getting DUX4 target engagement? My hope is that there's going to be enough data that we don't have to continue doing those in the future.
Thank you. Kate?
In a very amazingly optimistic future, when there are lots of drugs available for FSHD, what is, especially with looking at Duchenne, like what is the general approach to, if you do have multiple drugs that work differently, and have different mechanisms of action as we learned about today. How do doctors really decide where to start? Like, what is the hierarchy for if there are treatments available? How do people decide what is?
So, am I the right, I think that's me, right? Good question. So it's a great question, you know, I think it's, our clinic appointments have gotten much longer in our clinic in general because of these types of scenarios, right? And so this is always something we dreamed about for some of our diseases that did not have treatments that we're starting to see treatments for. But you know, the whole 20-minute appointment is now a 45-minute appointment because it's a complicated discussion. You know, we part of it is educational materials. As treatments get approved, what we do in Calgary specifically is it's really hard if you have, say, 200 patients in a clinic and a new treatment comes out. It's gonna take you a year to get them through the clinic, which isn't really fair. So we've been hosting sort of town halls where we invite everybody. And we have 8 neurologists and we share what's going on and that there's an approval and this and that, and then everybody just prescribes that they want it. And then we book all the follow-ups. And so now that's not because we're super nice. That's because like, it's just, that's how we can manage the workload. But in the end, it's a, you know, even if there's 3 treatments, there may be reasons why 1 or 2 may not be suitable for a given patient or not optimal, or sometimes it's just choice. I had an ALS patient on 5 treatments because he was on a clinical trial and was permitted to be on the other 4 treatments. And, you know, and so I was really excited to see him every month just to sort of see how he was doing. But you know, and our job is to advocate for the opportunity to have the most choice possible. And sometimes the patient will say, well, what would you do? And it's hard to know, right, what we would do, but, you know, we can sort of give some experience of how other people, especially as they accumulate experience with a drug, to say, look, most patients feel this way about this drug and feel this way about that drug, right? So.
Maybe I can just make a comment on that in terms of business reality, right? So the minute there is one drug on the market for FSHD, what will happen from the drug company perspective is the definition of unmet need will change. So if I cannot address an unmet need, which is unserved by the current drug, like let's say Alte's drug, which is really cool, works for all patients all the time, there is no market, right? We have to then go and be disciplined about what is the unmet need. Take Ozempic, for example, taking the world by storm, right? Companies like mine are looking at the Ozempics of the world and saying, wait a second, 40% of weight loss is attributed to muscle loss. So there's a new unmet need we created through that. And so the chances of there being, you know, drugs on the market that had the investment to go do the exact same thing is pretty low.
I think I learned in the SMA community, right? The first drug that was approved was nusinersen, and it was an intrathecally delivered ASO. So, and this is given to newborns. And so at the time it was life-saving. But then you had a gene therapy come along and that was given IV. So, and that was probably maybe easier to deliver and had a longer active time span that it was active. Nusinersen had to be given, I think, every 4 to 6 weeks. And now there's a small molecule that you can do an oral suspension at home to take. So I think in that field now, it's the people doing the clinical research to say which drugs are most effective, maybe in which order, which combination. But Greg, I'd still argue, you know, I worked at Pfizer and they were successful because Lipitor was the 5th statin around. But there's always a market there. There's a benefit risk ratio and there's always going to be competition. So I think it's a good thing. There's always, even with any drug approved, there's generally side effects and liabilities. And even though it's maybe not an unmet need, there's still a market advantage for competition for better therapy.
Yeah, I was about to that in the context of unmet needs. So, you know, being able to dosage time, being able to reduce dosage time, being able to administer, you know, you think about the patient value formula, right? Which is, if you're gonna invest money into something, you wanna be able to make sure you provide sort of exponential patient benefit or exponential reduction in cost to the system, not just the cost of the drug. So, you know, it's a nuanced thing, but it's reality, and if everyone experiences reality being on the other side of the investors, it's like, they'll really on this.
Yeah, I would say from my experience, Spinal muscular atrophy is a good example, just because they've had this success and they have available treatments and they are reimbursed by government and there's very good access for particularly children. And this was a fatal condition up until recently, and now it is a treatable condition. And if we treat children at the right time, they are essentially normal. And so as Eva mentioned, the first treatment came out. Now there are 3, and there's a 4th that just came out. It's like a complementary therapy. What's interesting about SMA, we have 60 patients in Calgary total. If you compare to asthma, 1 in 10 Canadians have asthma, and you think of all the medications for asthma, the market size, so the business case for SMA is about 50% of the whole asthma market in terms of how much revenue it can drive. The problem is the treatments are so expensive. And so in a way, that business situation helps our community because it attracts all the innovators to do this work, to have the money to do the trial, to compete with each other, which is what we want, right? We want them to do great work. But then later, it makes it a bit more difficult to get access, right? Because then we're dependent on government or private insurance, et cetera. So all of these things sort of have the good side and then the challenging side, I would say. Any questions for Ryan or Amy online? I know you guys are online. It's always tougher to be outside the room. So no further questions. Maybe if I can, a few more minutes.
Yeah, okay.
Okay, so maybe I'll ask Ian and Ryan, knowing your expertise is much deeper on the Duchenne side, as you indicated, but maybe each of you could share, if you think beyond your own company and your own technology, we've heard today about all these different targets within FSHD. We've heard all about the different approaches. What gives you the most hope for the future of this community, I guess, in terms of what's happening out there? And again, maybe not necessarily a specific drug or otherwise, but just the overall in terms of scientific progress. How do you even think about that in terms of that? But Amy, did you want to go first?
Boy, I have to say the biomarker work, because I think that can really transform a lot of things. I think it will be super important for Avidity, but also for all of the companies that are trying to develop things that specifically target DUX4. And you can imagine, I used to work in osteoporosis, and so initially people would do trials and they would measure the number of like hip fractures. But then they had a really good biomarker. And so then when new drugs would come, instead of having to do a 3-year trial and counting hip fractures, they could just demonstrate that they were showing a similar reduction in the biomarker. And that made things go a lot more quickly. The trials became smaller, easier to do, and it just really opened up drug development. And I think, so I'm of course partial to the biomarker that we're working on, but of course there are others and that relate to probably muscle atrophy or muscle growth. And you can kind of just imagine, instead of having to do 1 to 2 to 4 year trials or whatever they would be, much shorter trials, and really trying to understand and a lot of the biology. And I think that would also even get to the point of, our combination therapies, do certain combinations make more sense, right? So maybe something that's changing how your muscle stem cells behaves works really well when you're also knocking down DUX4. But in the absence of that, maybe it's harder in FSHD. And that's just an example, Ryan. I'm not trying to pick on you, but you could imagine using these tools that aren't 2-year-long efficacy studies to try and understand those, to really try and figure out which therapies should be perhaps combined. So I think, I don't want to run before we walk. We need to get the first thing approved. And I'm, we were many people working very hard for that. But then I think, as we go beyond that, I think there's a lot of opportunity to have smarter and more streamlined designs, especially as we have more tools that we can use.
I guess I would just add to that I think it's a very exciting time that we're in right now, where we actually have this option to think about a future where we can be adding things together, and that there's multiple programs coming from different angles. Even when I think about Duchenne, right, we had a first run of products that got accelerated approvals, where we were you know, and this isn't a poke at anybody, but we're kind of squinting, looking for whether there was changes in efficacy. And now I'll look at Avidity in both in Duchenne and in the FSHD space, you know, where I think we really are seeing some really interesting biomarker responses that could be indicative of, you know, tightening fibers up or calming damage down. And so to think of the sort of next generation modalities making a real impact now, and then with a lot of new things on the horizon in different degenerative muscle spaces, and different ways of looking at the problem. I think this is always the answer when we look to things like cancer. We know we don't have one drug that we treat for everything, although there are a couple that are treated across the board. But we do have multiple different ways of approaching these problems and products that are synergizing. And I just think it's fantastic that that's a conversation now, where not too long ago there was really nothing, right? There was nothing being trialed and there was nothing being thought of in the context of an approval. And I think that that landscape is changing very quickly, and hopefully we can, you know, see some really exciting data come out from any number of the groups that are working in this space. And we certainly take the view at Satellos that we hope to be part of a toolkit, right? Just one element in the toolkit that can address one aspect of these degenerative muscle diseases, and hopefully work in combination with multiple other products that can attack these problems from a different angle. So it's an exciting time, and I hope everything can continue to go forward at the pace it is.
Thanks, Ryan, thanks, Amy, and thanks, Eva, thank you all for good job, guys.
Thank you.
Bye. Thank you very much.
Thank you. Bye-bye.
See you soon. See you soon.